All right, excellent. All right, so it's just past, well, it's well past 3 p.m., so let's go ahead and get started. Welcome everyone to the second Marine Science seminar of the semester. It's great to have everyone join. I'm going to jump right into it and introduce today's speaker, Dr. Matthieu Leray. Dr. Leray is a postdoctoral researcher at the Smithsonian Tropical Research Institute in Panama. His research is focused on using broad-scale environmental sampling and manipulations of model systems to broadly understand the nature and causes of biodiversity patterns in the ocean. His research further seeks to quantify how biodiversity patterns influence ecosystem function and how ecological interactions promote the stability and resilience of marine communities. Dr. Leray completed his master's and PhD at the Université Pierre et Marie Curie. I don't know if I said that right, in Paris, France. And afterwards, he has received several postdoctoral fellowships, both from the Smithsonian and the Gordon and Betty Moore Foundation. Dr. Leray has over 40 publications with several in leading journals, such as the Proceeding of the National Academy of Sciences, Nature Communications, and Molecular Ecology. And lastly, I'll mention that he recently received a $1.6 million grant from the Mark and Rachel Rohr Foundation to study the drivers of coral reef resilience across the tropical Eastern Pacific. And so today, Matthieu is going to talk to us about drivers, function, and evolution of marine host-associated microbiomes across the Isthmus of Panama. So thank you, Matthieu, for being with us today, and take it away.
Yeah, thank you very much, Justin, and thank you, Jeremy, for inviting me too. It's really a pleasure to talk to you today, and I'll be mostly talking about what me and my colleagues have been working on in the past, I'd say 2.5 years. So it was only started this project about a year and a half before the pandemic started. And it's a project that's been funded by the Gordon and Betty Moore Foundation. And this is in collaboration between UC Davis and Smithsonian Tropical Research Institute. So I'll be talking about the drivers, function, and evolution of marine host-associated microbes across the Isthmus of Panama. So as some of you may know, the microbial symbionts, they fulfill for some hosts essential functions. And these functions have been pretty well studied in a few model systems. For example, these two. In the case of the Hawaiian bobtail squid, the squid is able to capture, acquire a microbe from the seawater around it. And it has special organs in its skin to essentially culture this microbe, and this microbe produces bioluminescence that it then uses to avoid predation. Another example is the gutless marine worm. The gutless marine worm, they live in sediments. They actually have almost entirely lost the ability to digest food themselves, and so they rely on several chemosynthetic bacteria to do that. Some ecosystems also rely almost entirely on microbes. The case of, for example, the coral reefs that you're very familiar with. The coral reefs are built by a nutritional symbiosis between corals, zooxanthellae that we consider microbes because they're unicellular, and also many other types of microbes. So the zooxanthellae provide some nutrients to the host, and the host builds the calcium carbonate, and then that benefits the entire ecosystem. There are many other microbes that actually are living in association with corals, among them fungi, archaea, viruses, and bacteria. For actually most of them, we have no idea what the function is. We can predict the function looking at genomes of these bacteria and archaea, but actually studying the function and understanding the function that they play for the host is really hard. This is another example of an ecosystem that relies on symbiosis, so interactions between host and a foundational host and microbe. Seagrass, that they have many types of microbes. Among them, they have nitrogen-fixing bacteria on leaves and roots that are helpful. And this is another case of where microbes that can be useful. They can influence biochemical cycling within ecosystem. For example, some sponge symbionts, they play a role in marine phosphorus and nitrogen cycles. It turns out, like with colleagues, we organized a workshop in Panama where we try to review as much of the literature as possible on the role that host-associated microbes play in ecosystems. And so if you want to know more about this particular field, you can read this paper that we published a couple of years ago. So there's a big question out there, is how have these interactions between host and microbe and hosts and microbiomes evolved? And this paper by Foster and colleagues in 2017 in Nature talks about the problem of a diverse microbiome. How can you evolve interaction between a host and multiple microbes that potentially compete for resources on a host with each other? I mean, it doesn't make a lot of sense evolutionarily. And so it remains a big question. And so what Foster and colleagues did is that they try to define broad categories of host microbiome or host microbe interactions. They try to classify these interactions in different categories. They, for example, talk about the systems where the symbiont controls the host. It's the case, for example, of hosts that have Wolbachia. Wolbachia will have an effect on the life cycle of the host. The system where the host controls one or a few bacteria is the case of this that has it interacts closely. It has an intimate relationship with a Vibrio, and there's another category of hosts that keep their microbiome on a leash. So these are hosts that have a lot of different microbes interacting with them, and they provide some kind of resources or some kind of habitat for the microbes. And in exchange, these microbes provide some kind of benefit or not to the host. And they talk about open ecosystem. Open ecosystems are hosts that have microbes, but the microbes don't really do much in the sense of a benefit or a lack of benefit to the host. So it's a simple way to kind of look at host microbe interactions. I find it quite useful. And it turns out in the ocean, you rarely find cases of symbiont control, and you rarely find, actually, there's no evidence of any host considered an open ecosystem to date. So most of the systems we know, most of the hosts interacting with microbes fall into host control and microbiome on a leash. So hosts that do control one or a couple of microbes basically fall into these two cases that are represented here on this graphic. You have either hosts that acquire their microbe or the microbe they closely interact with at every generation from the mother. So it's a vertical transmission of the microbe. This is the case of, for example, flatworms that live in a sediment. And you have, in other cases, you have hosts that would acquire at every generation the microbe from the seawater or the sediment around them. So the larvae are aposymbiotic, and they get the microbe when they basically settle. But these kinds of systems, the host control, are actually not very common. And there are cases where the host symbiont dependence is really high. The vast majority of hosts that are out there are hosts that have a lot of different microbes on their tissue and inside their tissue. Some of them have mixed mode of acquisition, so they will vertically acquire microbes from the mother, or they will get them from the environment, so they do both. And a lot of different species, actually the vast majority of species, get all of their microbes at every generation or from the sediment where they live. If you look at the research done on host microbe interactions, actually most of the research is highly biased towards a small number of hosts and microbes, mostly falling into this category of host control. So these are an example I've already shown, the gutless worm, the Hawaiian bobtail squid, but a couple more, the Lucinid clam, which I will talk about more in detail soon, and the sheepworm. And also a lot of this work is done on the effect of the microbes on the host, but not so much the host on the microbes. The microbiome, the systems that have keep the microbiome on leash are actually the vast majority of diversity in the ocean. There are a lot of studies now done on corals and sponges mostly that fall into this category, but a lot of these studies lack evolutionary and ecology context. This is where the Isthmus of Panama comes in. So the Isthmus of Panama basically provides a very nice context. And for those who are not familiar with the Isthmus formed about 2.8 million years ago. This is when it last closed. This bridge last closed. And about 3 million years ago, South America and North America were actually completely disconnected. There was water flowing between the two continents, and because water was flowing, animals were able to cross freely between the two oceans. So there was connection between these populations on both sides were connected. When the isthmus formed, things changed a lot. You had populations of the same species that suddenly became disconnected on either side. The Caribbean, which is here in yellow, became low in nutrients. Tides became suddenly much more low, and the temperature became constant. On the tropical Eastern Pacific, on the other hand, which is in blue, the nutrient concentration is much higher. There are large tides, and there are variable temperatures, especially seasonal. So you have hosts that have been evolving for about 3 million years on either side in very different environmental conditions, and their potential symbionts have done the same thing. They've evolved with them. We know a lot about hosts that have been separated by the isthmus, thanks to decades of research at STRI and from people coming to STRI or working in Panama. We know a lot about the taxonomy, the ecology, the behavior, and the evolution of hosts in different groups. We know a lot in echinoderms with the work by Harris Lesios on urchins. In gastropods, we know a lot about fish with the work by Ross Robertson and Ira Rubinoff. We know a lot about bivalves, and what's not represented in this graph, that is also an important group, are the arthropods. And we know a lot about the snapping shrimps that have evolved independently on either side of the Isthmus of Panama. We also have a physical and biological monitoring program that has been running for decades now. So we know about the environmental conditions at least in the past 40, 50 years on both sides. So what this project I'm going to talk about is really capitalizing on is all this knowledge about the host that has been accumulated for many, many years. And so we are interested in mainly 3 groups of hosts. We're interested in the fish in the microbiome. We're interested in the shrimps in the microbiome. These 2 groups fall into the category microbiome on a leash. We're interested also in the clams, which interact with chemosymbiotic microbe. This model group falls into the host control category. So before we started this project, we basically knew nothing about the microbiome of fish and shrimps, but we knew a lot about the host. For the clams, it was a little bit of the opposite. We knew a lot about the association between microbes and the host. We know a lot about actually what the microbes does for the host, because this group of clams that we're looking at, the Lucinid clams, they are widespread across the world's ocean, but we knew little about the hosts that were found on either side of the Isthmus of Panama. So the the limitations of both, of all these models were different, and the benefits too. So we're interested in several questions. We want to identify the ecological and evolutionary drivers of the microbiome. So what is the relative contribution of environment, host, and phylogeny and community assembly? We're interested in this aspect that has been looked in other groups. We're looking at phylosymbiosis. We're interested in looking at whether there's a relationship between ecological similarity or community similarity in the microbiomes and host phylogeny, which would tell us something about how those microbiomes evolved. We're also interested in co-divergence between host and microbe. We want to know which microbes have had long-term interactions with host. Are they mutualist? Are they parasites? This is particularly the case for fish and shrimps, for which we know literally nothing. We knew literally nothing about the microbiome, and how has the gene composition of the microbial genomes changed over time. And we're also interested in adaptation and metabolic cooperation. Do we see evidence of adaptations in microbial genomes that are separated by the isthmus, and to what selective pressures are they changing? And is the metabolic function of the microbe evolving, and how? I'm not going to talk too much about this particular aspect of the work because we're still working on it. But if you invite me in another year, I'll probably have a lot more to say about this. So this is a representation of how these fish system looks like. You basically have many different fish families, or representative of many different fish families that have been separated by the Isthmus of Panama. In many cases, you have species that have been, for which the separation has been dated to be around the time the isthmus closed, so about 3 million years ago. And you have outgroups for each. And each of these fish, a lot of these fish fall into different trophic groups, which allows you to test how microbiome evolved. For example, gut microbiome and skin microbiome evolved in different trophic niches. So this is how the phylogeny of the butterfly fishes that are on both sides of the Isthmus of Panama look like. In yellow, you have the species that are in the Caribbean side. In blue, you have the species that are in the Pacific side. You see here in orange, the line here represents the closure of the Isthmus of Panama. So you've literally, right when the isthmus closed, these two species separated, Chaetodonocelatus and Emerald separated. And right before the isthmus closed, you have a speciation on the Caribbean side with these two species that are that diverged. So we're looking at different tissue types to look at what the microbiome is made of and how it changes across ocean and across species. So this is a representation of this for the gut microbiome of this butterfly fish. So each color represents a species in this graph. The circles and the triangles are individual fish or individual gut microbiomes, and the circles are for the Caribbean species and the triangles are for the Pacific species. And as you can see here, there are different gut microbiomes on both sides, but there's actually also a lot of similarity, which is interesting. Many different microbes, at least looking at 16S, which is maybe not the best marker for this, are shared across the isthmus. So the Caribbean and the Pacific species have identical 16S ASVs. 16S doesn't evolve very quickly, so it could be due to the resolution of the gene. So we're definitely looking at now taking the next step and looking at metagenomes for a subset of these fish. But what we're looking at is 11% of the variance in community composition is explained by ocean and 23% by species, and 66 is residual. So there's a lot of variance that remains unexplained, but about 10% is explained by ocean. So there are limited differences, I would say, between oceans. We looked at phylosymbiosis. So in the left side, you have the host tree that I just showed you. And on the right side, you have tree that is based on the community similarity of the gut microbiome. And so if the fishes are grouping together, it means the gut microbiome is very similar. And what you expect if microbiomes is related to, for example, some kind of trait of the fish, for example, if ecologically or phylogenetically related fish eat the same thing, you expect to see a very similar microbiome. And as you see, it's not at all the case. We see here the two sister species that don't group at all together on these UPGMA tree based on the gut microbiome. So there's no evidence of phylosymbiosis in the fishes we're looking at. Not only in butterfly fish, but other species, other groups of fish that we've looked at, we haven't seen evidence of phylosymbiosis. We're interested in cophylogeny. Are some bacterial trees actually, or some bacteria co-evolving with fishes? If they do, we should see the two trees, the bacterial tree and the host tree, sort of match each other, and we've looked at this for many different bacterial genera for a bunch of different fish. This is a representation of how these cophylogenies look like. You have connections between this particular microbe and this particular fish. It turns out here, this particular fish, Chytodon striatus, has many microbes across the whole bacterial tree. We looked at cophylogeny across a bunch of bacterial genera, and we found significant cophylogeny with a single group of bacteria, the group Endozoicomonas, which is interesting because Endozoicomonas is known to be a symbiont in many other groups, including in fish. It's been shown to be linked with disease, but also in sponges, tunicate, hydrothermal vent snails, and polychaetes. So it seems to be a group of bacteria that has evolved to become a symbiont with many, many different host types. And interestingly, in a few studies that I've seen looking at cophylogeny between host and microbe, in the most recent on corals, they show that Endozoicomonas has co-diverged with corals, which is really interesting to see. Now I'm going to switch to a different host type, but doing this very similar analysis. So we are also looking at snapping shrimps. Snapping shrimps have been heavily studied by Nancy Norton and her group. And recently, colleagues Carla Hurt and Kristen Hallgreen published a backbone host tree of the snapping shrimps. These don't only include the Panamanian species, they include species from across the tree of that group. But we also have, we can also capitalize on previous gene trees to really resolve the tips and understand which of the snapping shrimps are sisters to each other and for how long they've been sisters. It's an interesting group also because it's one of the few groups where staggered evolution has been shown. So as the isthmus formed, there was obviously water flowing across the isthmus until probably the last minute. And the species that typically live in deeper habitats separated first, and species that live in shallow habitat, mangrove habitat, separated last. So it's a nice gradient of separation time. So we went across the country, took about a year and a half to do this. And we collected the snapping shrimps that Nancy Norton and Arthur Anker and others have worked on for 40 years, or 30 years. It took a lot of work because not only are they hard to find and catch, but also they're hard to identify. So here is Arthur looking through a sieve. He probably found something interesting in there and he's identifying it. And we know this is actually a lot of people helped with these collections. And these are some of the habitats where we find them. A lot of them live in burrows, so we have to suck them out of the burrows under rocks, and some of them live in association with other hosts, or live actually in the cracks formed by rocks. So we have to break rocks, and they live in little holes in there. In the process of recollecting all these species, we actually found many new taxa. These are some examples. A lot of them come from the tropical Eastern Pacific, which is an area that's a lot less studied than the Caribbean. And so we found so far described 2 new genera and 8 new species of shrimps and bivalves. So we focused the microbiome analysis on 27 species across 6 major clades. And here, the yellow rectangles, they show the sister taxa. And here you can see photos of the Eastern Pacific and the Caribbean sister. They actually look very much alike. And if you go to a habitat where you found the Eastern Pacific sister, you look for it in the same habitat in the Caribbean, you're gonna find it. They have conserved like the same behavior and habitat preferences. We dissected 5 different tissue types. We looked at the gill microbiome, hepatopancreas, eggs, stomach, and mid gut for a total of 426 specimens. And we did a similar analysis as I just showed you for the fish. We looked at whether there was evidence of phylosymbiosis. Again, no. Actually, this is the mid-gut microbiome, and for all tissue that we looked at, there was no evidence of phylosymbiosis. So here they seem to group more by ocean, which is not, but it's not nearly as clean as we would think. We also look at cophylogeny. We tested cophylogeny between a bunch of different bacteria and host tree. And again, we found a single group of bacteria that shows significant cophylogeny with the host. It's the genus Vibrio. And Vibrio, as you may remember, is also known to be a symbiont, for example, for the squid. So there are clearly important microbial symbionts that have been identified, and again that we showed that are co-evolving with hosts, that are interesting groups to look at in the future in terms of how they interact with the host and what they do, and looking at how they've evolved. Now switching to the last group of hosts, the Lucinid clams. This is work that's conducted in collaboration with Laetitia Wilkins, who was at UC Davis, but now is got a job at Max Planck in Germany, and Gillian Petersen at the University of Vienna. The system is really cool. So each clam has a single sulfur oxidizing symbiont that is actually in very high density in its gill. Here, in this image, you see it's a microscopy on the gill. In red, you see the symbiont cells, and in blue, the host cell. So it's actually many more symbiont cells than actual host cell in the gill. This is actually quite nice when you work on microbes, because it means that you can just shotgun sequence the whole gill tissue, and you get many sequences of the microbe, and you can actually assemble the whole genome pretty easily. They are found in sulfur-rich sediments under seagrass, and the symbiont is acquired by the settling larvae from the sediment or the seagrass itself. We actually don't know. The larvae don't have the symbiont, that we know for sure, but we don't know where or how they get the symbionts from, when they settle on the seagrass or on the sand. It's an interesting group because actually it was hypothesized that they've been really fundamental for the spread of seagrass habitats around the world, because seagrass really don't like sulfur, and because the host and the symbiont remove sulfur or lower the concentration of sulfur in the sediment, they actually make the conditions much more suitable for the seagrass plants, and then for the rest of the the diversity that lives in seagrass. This is a tree of the group, or the Lucinid group, based on morphological characters, I believe so. And we actually focused on two different genera, the Kodachia here in the top that has sister species. The two are separated by the isthmus, and Tina. In this analysis on the microbiome, we also included Clatrolucina, which is a very common Lucinid clam on both sides. And so we collected it because it's probably very ecologically important for seagrass. We actually, for this group, we were able to expand the sampling much more than just focusing on Panama, because it turns out there are many specimens preserved in ethanol in museums. And Gustave Paulé at the Florida Museum of Natural History had a really nice collection of Lucinid clams from a lot of different locations. So we actually able to cover the Caribbean and many locations in tropical Eastern Pacific. And we actually extended that sampling to the Eastern Atlantic and the Indo-Pacific. So, and I'll show you some of those results. As I say, we can dissect the tissue or the gill, and we can actually shotgun without any complicated manipulation. We can extract the DNA, shotgun sequence. We cut the DNA in pieces and then sequence it, and then assemble the whole microbial genome for every clam, which is very, very convenient. This is a slide of results. So here you see this tree is based on 233 high quality genomes that clustered into 9 different clades. So these are the 9 clades you see here, the triangles. And the distribution here you see in yellow are the Western Atlantic samples coming from, so the symbiont coming from Western Atlantic host. In blue, you have the tropical Eastern Pacific samples. And in orange here, you have Indo-Pacific. So what you can immediately see is that there is a nice separation between Western Atlantic and tropical Eastern Pacific. What is also quite clear when you look at the host where these symbionts were found is that many of these symbionts are found in many different hosts. They are not host specific at all, at least for the majority of the ones we look at. Laetitia called these symbionts promiscuous, which I don't know if is a good term, but at least it makes it clear that they're not specific to one partner. One interesting observation from this tree is that there is one symbiont that seems to be particularly promiscuous. It's found in many different hosts, and its distribution is remarkably large. This is a map of where that symbiont was found. Here in pink, you see this particular symbiont that has a wide distribution where it was found, all the way to Australia, here in Africa, Eastern Atlantic, and Caribbean. It has a true cosmopolitan distribution. It's the only one that we found with such a large distribution. The other ones had more restricted distribution. This one in green is limited to the Caribbean. You have Mediterranean here, and this one is only found in Australia so far, but probably more locations if we keep sampling. So how it maintains the genetic homogeneity across this huge distribution is a good question. We found high rates of recombination in the genome of these particular symbiont, much higher than in other symbionts. So it's one possibility, but it's really interesting, and we tentatively named this cosmopolitan symbiont after John Taylor, who has spent his entire life working on this group of Lucinid, and without which we wouldn't be able to do this work, because he's done all the groundwork on the host. So now I'm going to switch to something you're probably thinking is completely unrelated, but actually it's not, and I'll explain why. I will talk about a little bit ocean deoxygenation. So ocean deoxygenation is definitely an emerging threat. And by the way, I want to mention this work that I'm going to talk about has been done in collaboration with William Weed, who is at FIU, working with Justin. So thanks, Will. And it's clearly a threat. And if you look at this map, it's pretty concerning. This map was done in a paper by Andrew Altieri and colleagues in 2017. And it basically looked at where dead zones, tropical dead zones, were across the world ocean, and coral reefs are in more than 50% of known tropical dead zones. So if we think about ocean deoxygenation affecting coral reefs more and more in the future, we can think of potentially alarming consequences on biodiversity. These events of deoxygenation or hypoxia are actually really hard to notice, because they usually happen within they only last a few days. So if you're not there looking, you likely miss them, and you'll think that reefs are gone because of some unrelated reason, like potential bleaching due to thermal stress. But in 2011, turns out, by chance, Nancy Norton was working in Bocas del Toro when one of the hypoxic event happened. And what she saw, she was diving on a reef close to the station. This is a map of the Bocas del Toro, or Bay of Almirante in Bocas del Toro. She was diving on one of these reefs next to the station when she noticed that some sponges were half dead, half alive. And the live half was the one above, and the dead part was the bottom part of the sponge. And she wondered why, and she immediately thought about something, some kind of zonation in the water column. And so she went out with sensors, and she measured chlorophyll, temperature, pH, and oxygen. And this is when they realized there was low oxygen water really coming shallow, coming from the depth, coming to the reefs, and killing the corals and everything else. So this happened in 2011. This was documented, and in the meantime, in the following years, we actually, not me, but others, and then when I came in, I was involved in that research, sort of prepared for really capturing what was going to happen in a subsequent one. And in 2017, when the other deoxygenation hypoxic event happened, we were actually prepared to have data before, during, and after. So this hypoxic event happened in a different area of the bay, more in the south, and actually the low oxygen water came even shallower than in 2011. So killed corals and other species and other individuals that hadn't been affected in the 2011 event. This is what Will, who actually was part of that team, Maggie Johnson, Noel Lucia Rodriguez, saw when they went out that day. They saw animals that you usually don't see on a reef coming out of the coral and suffocating, literally, like just trying to look for air and then eventually dying. So what we did, by we I mean Will and Lucia and Noel, what they did was go out and document by collecting samples what was happening to the corals and also to the microbes during this event. And we had data from before, and I did it after. So we first look at what was happening to corals exposed to hypoxic water. And we've had, it's not very surprising to show that, yeah, they were deeply affected by hypoxia. We looked at Agaricia, the lettuce coral, and a lot of them bleached during this event. It turns out that actually not all corals are affected the same way. Some corals were actually able to cope with low oxygen water for a few days, others didn't. Agaricia is one of them. So we clearly showed it looking at these bleaching descriptors. This is how the reef looked like during the hypoxic event. It looked like this a year after, and this is how it looked 2 years after. So clearly an event that only lasted 4 days, very long lasting consequences. Some of these reefs never recovered, they turned into sand, and the coral composition, the benthic composition changed drastically on the reefs that were affected. What I'm going to talk about now a little bit is the microbes, since this is the main topic of the talk. We actually were able to measure what happened to the microbes that were around the coral, so the free-living microbes that are around the corals and the sponges during this hypoxic event, and after and before. And this is a very complicated representation of the microbial community, but basically these circles are genomes, microbial genomes, and the orange is hypoxic sample, so sample collected during the hypoxic event, and blue control samples, basically normal water, normal oxygen concentration after and during at a different site. I'm not going to go into details in this graph. What I want to show you is that we found, interestingly, one species of microbe that thrived at really low oxygen. It's a type of microbe called in a group Poseidonibacter, and this is a representation of its entire genome. So it's reconstructed from shotgun metagenome. So we assemble the pieces into different genomes. And this group has been fairly well studied in other systems. So we were able to take that genome and download a bunch of different genomes from GeneBank that are from related species in that group, and look at where else and these genomes were found, are actually the ones that are affiliated with the one we found during the hypoxic event, where they were found, and if they were on what kind of habitat they were found. And to our surprise, turns out this microbe is likely host associated. It had been found in association with scallops, abalones, and other hosts, which to us suggests that it's normally a microbe that lives associated with a host, or that's part of the benthic community. So it really thrived in conditions in its free living stage in conditions of hypoxia, when normally it is when it also has a host associated stage. So what does it tell us? So this is a representation of the same representation I showed earlier. So we used to think about, when we think about host-associated microbe, we used to think about how they acquired by the host, but we never think about the other way around. So these host-associated microbes, when they are in the free living stages, we actually have very limited understanding of what their function is, where they are found, and what exactly they do. And so it's possible that there's also these microbes could go when the hosts are dying. They could go back to the free living stage and thrive in conditions like we found in Bocas del Toro. So it really got us to think about how do we understand this feedback between host-associated free-living microbes and environment, and how they affect the host and how these associations evolve. And so what we turned out to do is we write a piece where we basically expose our vision of how we could do that. And our idea is that basically we need to change a little bit the way we fund science and the way we do science on host associated microbes. We are typically thinking of we're typically doing science that is pretty short term, and we think that really to understand these complex processes, we need to take this short term research mostly on individual study systems, and a lot of this work is done in aquaria into the field, and we need to do long-term research on this system. So we proposed to set up observatories of host microbe ecology and evolution in particular places where the ecology and evolution of host is fairly well known. Obviously, Panama is an interesting place. So this is how we would do it, like sample through time, selected hosts in their natural environment and also their surrounding habitat, and look at different processes within those microbiomes. Obviously, there are many other places that would be really interesting for this kind of focused work over the long term. We're thinking, for example, of emerging islands. We're thinking of CO2 vents in Italy. We're thinking of urbanized area like Hong Kong, places like this where we know a lot about the environmental conditions. And as I said, really the idea is to take this research on host microbe interactions from the short term, which is useful because it allows us to really understand the nitty-gritty of the interactions and how some of these microbiome and hosts can acclimatize to changing conditions to the long term. So we can look at how these interactions become altered, and eventually how it turns into genetic adaptation, both in the microbiome and in the host. So this is all I have for today, and I want to thank all my colleagues, many of them. Again, thanks Will, thanks Andrew Altieri, and many others who worked with me in Panama on this project. And I'll take any questions you have. Thank you very much.
Great, thanks Matthieu, thanks for a great talk. We have a pretty small group today, so if anyone has any questions, you can just go ahead and speak up and ask our speaker.
Matthieu, a very good talk, and I appreciate you putting Alpheus milziae on your phylogenetic tree.
You're welcome.
I have a question about the hypoxic event. I guess I should know this from speaking with Maggie, but is this, have you guys figured out what the trigger was for that particular event? Is this something that occurs seasonally? It happens every year, or is there some like broader forcing that happens that triggers some of these hypoxic events in like 2011 and 2017?
Yeah, it's not entirely clear yet, but I think it's a combination of a couple of things, really hot days, and absence of wind. So really stagnant water. Yeah. So there's a thermocline that forms, and then you have all that. The bay of Bocas is known for having this really low oxygen water in the bottom. Deeper, and for some reason that in some areas it just comes shallow.
Got it. And sort of like a related question, you mentioned, and perhaps this was sort of what Maggie was looking at, you mentioned some species of corals were somewhat resistant to the hypoxic event. Has that's something that you guys have been looking at? Do you guys have any indication of why certain species were resistant to some of the hypoxia?
It's a good question. I'm actually, I think this is what Andrew and Maggie are looking at now.
Yeah.
Interestingly, yeah, some of the massive corals seem to be really doing okay, like Orbicella. It's not clear what the mechanisms are.
Yeah.
At all. That's what they're gonna hopefully find out soon.
Got it. Thank you.
Yeah.
Maggie Johnson actually came and gave a marine science seminar here at FIU about the deoxygenation.
You knew everything about it already.
Yeah, it was in our the last academic year.
Wonderful.
I have, yes, it was really a good talk too, a great talk. I have a question. I was just wondering, you know, what's known about how, you know, for example, you mentioned that the microbial symbionts can be acquired from the environment, or there can be vertical transmission, you know, during reproduction. And I'm wondering if what's known about the evolution of or the evolution or the circumstances under which acquisition from the environment versus vertical transmission occurs. Does acquisition from the environment eventually evolve into vertical transmission, or is it, you know, is there a lot of flexibility that's maintained? You know, what's known about that?
It's a tough question. Yeah, so I wouldn't be able to say if vertical transmission evolves from horizontal transmission. That would be my intuition would be the case. What I can tell you is that one pattern that's quite interesting is that in the ocean you have a lot less vertical transmission than on land, and you know, and some have hypothesized that is because if the symbiont can disperse, and if it's easy to acquire from your surroundings, why would you vertical transmission doesn't evolve as often. It's vertical transmission is also I mean, it can be is not. So there's a literature on insect microbes, microbes associated with insects, about how the genome reduction in vertically acquired microbes, that it's not evolutionary as stable as horizontal transmission, because horizontal transmission allows this host to really exchange and pick up the microbes, the symbionts that are more adapted to the environmental conditions where they are, and so it gives them more flexibility to respond to changing conditions than vertically transmitted microbes do, and it could be also why the vertical transmission is not as common in the ocean. So how that they evolve, I think is probably group specific, and so I don't know if there's much on it.
Well, certainly there's a good argument to be made that that's why you have, you know, horizontal transmission, acquisition from the environment of the Symbiodinaceae in corals, because, well, you know, they can settle in under different conditions, and they needed a strain or a species of endosymbiont that's adapted to, say, higher temperature or something like that. And there's actually been quite a lot of research recently that's been published on that.
That's true. Yeah.
Mm-hmm. Yeah.
Absolutely.
So that's a really good point. Thank you.
Yeah.
Great. Any other questions? All right. Well, thanks, Matthieu, for giving us a talk. We really appreciate it, and we'll have to invite you back for another talk. And I hope to see you next year in Panama.
Yeah, for sure. Let me know if you come by.
Yeah, we'll do.
Thank you very much.
Thank you very much.
Thank you, Matthieu.
Really an interesting talk. Really.
I'm glad.
Thank you. Really enjoyed it. Thank you.
Bye, everyone.
Bye.